the chair of the Medical Advisory Board for the DYRK1A Syndrome Foundation. I am joined here today with Doctors Anna Falzer and Dr. Kellen Weston. They're both at Combined Brain. Dr. Falzer is the Chief Scientific Officer, I think if I got that right, and Dr. Weston is a postdoc there. And although this may be the first time that you're seeing them, they've been very instrumental in helping to support our research objectives at the DYRK1A Syndrome Foundation. And they've been in the background for a while. I think Kellen was at the family meetup presenting, but they've been so instrumental in moving things forward. And we decided that instead of just loading everybody with research for one time a year at the family meetup, we thought it would be nice to kind of break this up over the course of the year. So the idea is to have these research roundtables quarterly. But if there's enough interest, we can try to maybe increase the frequency or offer, other opportunities for researchers to present. So I think after this is all said and done, we'll go ahead and send out an email. So if you have any feedback or if there's any research topics or questions that you may have, feel free to respond to that. So without further ado, I'll pass it on to Dr. Weston, and we're going to be covering the role of genetic variants in DYRK1A syndrome.
Hi, everyone. Nice to, virtually meet you. So today, the title of our research roundtable is Understanding the Role of Genetic Variants in DYRK1A Syndrome. And there we go. Okay, so I will be giving part one of our roundtable today, and then Anna, Dr. Falzer, will be giving part two. I will be talking to you all about genetic reports and kind of covering what they are, how to read them, and why do they matter. So when you come into the clinic and your doctor suspects that the symptoms that you came into the doctor's office for might be caused by genetic anomalies, your doctor might then go ahead and order for you to get a blood draw or some other sort of tissue sample, like a cheek swab, for example. And then that will get sent to a genetic testing laboratory where they will isolate your DNA and test for DNA mutations. And these testing laboratories can be associated with your hospital, or they can be sent to an outside company like GeneDx, Ambry, Baylor, Invitae. These are really common companies that a lot of your genetics may have been done at. Also, in more rare cases, if you are participating in a research study, it may have been a research laboratory that did your DNA testing. There are several different types of genetic tests that many of you have gotten for your children. So two types are whole genome sequencing, and that tests all of your DNA. You can also have whole exome sequencing, and this is much more common. I've seen whole exome sequencing done for a lot of the genetic reports that I've seen in the DYRK1A community. And whole exome sequencing tests for the parts of DNA, basically, that contain genes that code for proteins. It does not test for all of the regulatory DNA that doesn't code for proteins. You can also get a gene panel done. This is very, very common as well. In a gene panel, it tests for a predetermined set of genes, which can be anywhere from several genes to hundreds of genes. So for an example, if a patient comes into the clinic with epilepsy, their doctor might put in an order to the genetic testing laboratory to do a comprehensive epilepsy panel. And often the epilepsy panel is about several hundred genes that are known to be associated with epilepsy symptoms. You can also get single gene sequencing done that only tests for one gene. That's a little bit rarer, but it might be more likely if you did participate in a research study. So after you get your genetic testing done, you receive this genetic report. And I don't know if you guys can see my mouse, but this here is an example of a genetic report. It's given to you by your clinician, your geneticist, or your genetic counselor. It's about 2 to 6 pages in length on average, and it summarizes the genetic testing results. So it tells you what kind of sequencing you got, whether it was a whole exome sequencing or a gene panel, etc. It gives you your results. So it tells you which gene is thought to primarily be causative for your symptoms. So in this case, in this example, this person had a mutation in their P10 gene. And also, I'll talk about this later in this presentation, but it will also give you your interpretation and your secondary findings, which is other mutations that may have been found during this screen. And if you didn't receive a genetic report, you can usually ask your doctor to send it to you, send you a new copy. It's a pretty simple process, depending on where you got it done. And I've had to re-request copies myself before. It was pretty simple. So often people get this genetic report. Again, it's a bunch of pages full of dense language, and they really wonder what it all means. You might see the gene name DYRK1A here, but other than that, you may not know how to read this otherwise. So to really understand your genetic report, we've got to take a step back and just cover some basic molecular biology. So how genes, proteins, and mutations work. So to use an analogy, if you think of your 23 pairs of chromosomes, and you have a copy of all of these chromosomes in every single cell of your body, if you think of them as a stack of recipe books, in that case, then a gene might be considered a specific recipe. So in this case, the recipe for chocolate chip cookies. And DNA is made up of individual building blocks. They're called nucleotides. And 3 nucleotides together is called a codon. And so a codon might be counted in this example as an individual word in the recipe. So eggs, flour, vanilla. And then one codon is read by your cell's machinery to be translated into a protein. So each codon, your cell will read this instruction manual and say, okay, this codon codes for one building block of a protein. And it'll read through your codons on your DNA and slowly build this chain that once gets folded together and packaged becomes a protein. So that would be your final product in this example, a chocolate chip cookie. So where a lot of parents especially ask where gene mutations come from, there are really 2 main types of gene mutation. So basically, where can this recipe process go wrong? And there's 2 broad categories for that. The first is a de novo mutation, and all of DYRK1A mutations that have ever been found before have been de novo mutations. It literally means from the new, and these are mutations that happen spontaneously. So these can happen in the sperm and the egg, or in rare cases, depending, very, very, very shortly after conception. And so the risk of a second child with the same mutation in this case is extremely low. You can also have inherited mutations. So far, no DYRK1A patients have had it inherited so far, just due to the nature of DYRK1A. These mutations come from one of the parents, and the parent may be a non-symptomatic carrier of that mutation. And in that case, any children of that parent with the mutation are at risk of inheriting it. And I just want to really emphasize that we think of mutations as this big, scary thing. But to be honest, every single person, including you, including me, does have some sort of genetic variation, genetic mutations of both types even. But really only some mutations can cause genetic disorders. So now that we've covered genes, back to genetic reports and how to read them. So going back to our example here, this was a GeneDx report, and this is their exome DX. That doesn't really matter as much as that it tells you that the test that was requested was, the type of test was whole exome sequencing in this example. It'll tell you the clinical indication, which is the reason that your doctor ordered the test. So in this example, this patient was experiencing seizures, which is what caused the doctor to put in an order to GeneDx to get DNA sequencing. It'll give you your test results. Test results usually, in most of the cases I've seen, come in this table format, and you might actually have multiple test results. So in this example, it's just a kind of generic example. This person has mutations in gene X, gene Y, and gene Z. So their primary mutation may be in gene X, but Y and Z are their secondary findings. And we'll talk more in the next few slides about what each of these columns means and the importance of secondary findings. And then finally, you have your interpretation, and that's kind of what takes up a lot of the bulk of your genetic report. And your diagnosis information is here. It gives you information on the gene, on the mutation, on previous research on the mutation, And if there have been any reported symptoms associated with that mutation, they'll tell you about it in this section. So to read a genetic report, here's a table which basically represents most of the different boxes in that table on your genetic reports that you might see. So the simplest to understand is the gene and the disease. So the gene in our case would be DYRK1A, and DYRK1A is this enzyme. It's dual specificity tyrosine phosphorylation regulated kinase 1A, which is quite the mouthful. And the disorder it's associated with is DYRK1A syndrome. DYRK1A syndrome did used to be referred to as MERD7 in some contexts. And so you may or may not, especially if you have a very old genetic report, there's a chance you could see that, but it's fairly uncommon. Mode of inheritance is something that I think that a lot of people get confused about. So in this case, in this example, the mode of inheritance is autosomal dominant, which is what I think pretty much everyone with DYRK1A is going to see. So autosomal basically just means that the mutation is not on the X or Y chromosomes, which makes sense because DYRK1A is on chromosome 21. It's not on your X or Y chromosomes. And then dominant means that you only need one copy of that disease mutation to cause the disorder. On the other hand, recessive means that you need to have that mutation on both copies of your chromosome 21. Because again, each chromosome, you have one copy from mom, one copy from dad, and so you only really need one copy to have a mutation on it, whether it's your mom's copy or your dad's copy, to have DYRK1A syndrome. Zygosity and inherited from. So zygosity is basically, this is kind of redundant. It's covering the same concept basically. So again, your chromosomes come in pairs. So chromosome 21, you have two of them in the pair, one from mom, one from dad. Heterozygous means only one chromosome in the pair has the variant. Homozygous means both chromosomes. I think pretty much all of you will probably have seen heterozygous on your reports. And then again, de novo means that the variant happened spontaneously. It was not actually, well, it's on either mom or dad's chromosome. It wasn't inherited from mom or dad. And then this classification is what a lot of doctors and researchers really focus on. So variants are classified in three major categories, either as pathogenic or likely pathogenic. That means that they are likely to cause disease symptoms. They're classified as benign. Actually, they can also be likely benign if they're pretty sure, or they can be classified as a variant of unknown significance, or a VUS. And this prediction is generated by a combination of previous research on that gene, as well as computer modeling to kind of predict what the severity of that, what impact that mutation may have. Okay, so now skipping back a little bit to the coding DNA section. This is the exact mutation that was found in the DNA. So I'm going to kind of walk you through how to read this. So in this case, the variant is called the coding variant because DNA is the code to make proteins. So here, the C basically just denotes that we're talking about the DNA. C stands for coding DNA. And then 1042, this means that it's the 1042nd building block or nucleotide in DNA that's mutated, in the gene that's mutated. And then this section here, this G, and then it's a little arrow to A, means that the correct building block or nucleotide at this 1042nd position should be a G, which is guanine, but it instead has been swapped out or mutated to an A, which stands for adenine. These are just different building blocks in DNA. So you could read this all together as, instead of a guanine, the 1042nd amino acid, this should say nucleotide, I can fix that, is mutated into adenine. And now I'm going to talk about the protein variant, because again, DNA is the instruction for proteins. So this section denotes what happens to the protein because of the DNA mutation. So it's a very similar notation. So here P denotes we're talking about the protein level. And then this G, this is one of the amino acids or building blocks. You can kind of visualize it as beads on the chain that build a protein. So this G denotes the amino acid glycine in this example. And then this is the 348th amino acid in this chain that makes up the protein. And this is an R, which stands for arginine in this case. So you could read this as, instead of a glycine, the 348th amino acid has been mutated to an arginine. And there are different types of protein variants you can get. There's different ways your protein can be altered. So the example that I just gave was a missense variant. This is where one amino acid or one building block of the protein has been swapped out for the wrong amino acid. So, G to R mutation. You can also have a nonsense variant. This is when the mutation in your DNA creates this stop sign codon. So it's an indication for your cell's machinery that's reading the DNA and creating a protein based on it. It's an indication to stop. So you essentially get a truncated protein, a shortened protein. You might just have the first half or even just the first little bit of your protein, and then it gets cut off. So this in your genetic report is going to be annotated as either with an asterisk or an X, just depending on who did your sequencing and how their annotations work. You can also have a frame shift mutation. This gets a little bit more complicated. I'm just going to say that there's usually a nucleic acid. So there's a building block in the DNA that gets inserted or taken out. And so again, DNA is in these triplets called codons, and your cell reads each codon as like a word. And then that word codes for an amino acid. So if you had, 9 DNA nucleotides, that would equate to 3 codons, 3 sets of 3, which would be 3 amino acids. But if you have one of those building blocks pulled out, you're going to get a frame shift. So your cell's machinery is going to be out of sync when coding for the protein. So this is going to be, and I can answer more questions about the biology of that if that's confusing. This is going to be annotated as FS for frame shift. And then you might have a little asterisk here with a number. And this just means that once the frame shift has happened, and then your cell machinery is now reading the wrong codon for every single codon. This is going to tell you when this train that's kind of gone off the rails stops, when that stop sign finally will come into your DNA. So in this case, this person has a frame shift that will cause the DNA to stop reading by about 425 amino acids. I'm not going to talk about this too much, but there are other types of mutations you can have that are splice site mutations or mutations in the untranslated region. These get a lot more tricky. I'm not going to cover them in this very basic talk. If you happen to have one of these mutations, I'm happy to talk to you about it. But if you have one of these, you probably won't get any sort of protein coding annotation in that box of your genetic report. Now I want to talk about secondary findings. Secondary findings are gene variants that the genetic test picked up that are not considered to be directly responsible for your primary symptoms. So secondary findings are important for DYRK1A patients because these mutated genes may have an interaction with the same biological pathways that DYRK1A affects. And if that's the case, researchers really want to know about those secondary mutations. And they could also contribute to more severe symptoms. But again, we don't always know, and it needs more study. And any extra variants might contribute to the broad range. Oh, that's kind of, I said that, but the broad range of symptoms in patients. So secondary findings in your genetic report are going to be often reported right under the sort of main gene. So in this example, the main gene is X. So Y and Z would be your secondary findings. In this example here, this genetic report put this person as positive for a DYRK1A mutation, and that's classified as pathogenic. But they also have mutations in these other 3 genes that are of uncertain significance. And here's a couple more examples. In both of these genetic reports that I pulled, both of these tables were on page 4 or even 5 of the genetic report. So this is why the whole genetic report and kind of looking at the whole thing holistically really matters, is that you may have a lot of these variants that are reported later in your report. So a little bit about why do all of these variants matter? And that's because you have all of these very different types of mutations that all cause the same DYRK1A syndrome. So many different type of variants, but we all seem to have the same overall DYRK1A syndrome with a lot of very, very similar symptoms among our community members. So researchers really, really want to understand, how do all of these very different variants all cause the same disorder? And then do different variants cause different symptoms sometimes? And once we start developing therapies, different variants may need different types of therapies to be developed for them. So we are asking, DYRK1A, the DYRK1A International Alliance has partnered with the website, the platform RareX. RareX is a platform where you can upload, you can sign in, you can register as a DYRK1A patient. We can see where DYRK1A families are across the globe. We can also get a lot of information from you guys for researchers to be able to study to learn more about DYRK1A syndrome. So we're asking for those who have signed up for RareX or to please sign up for RareX to upload your genetic report on RareX. It's pretty easy. This is a video of that uploading process. Amy was very good to do this. So if you can notice, this video is only 29 seconds long, so it is very quick and easy to upload your genetic report. So in RareX, you have your documents. Oops, I can't point, can I? You have your documents folder. Oops, there it goes. And then you have a button for upload. You click what type of upload you're doing and click genetic test. Click the upload button, upload a file, go through your files, find your genetic report as a PDF or a picture, click save, and then it pops right up on your RareX account. And then the RareX team will take it from there and they will look through your genetic report and they'll take records of what kind of variant you had, and they'll take records of your secondary findings as well. And then that can go into more information for researchers to understand what kind of genetic variants DYRK1A syndrome people have. One side note is to ask if you do upload your genetic report, which please do, please upload your entire report. So here's an example, I've highly redacted it. A camera photo is perfectly fine. If you were handed a physical sheet of paper by your doctor, feel free to take out your phone, snap a quick photo of it. We're totally okay with that. But please snap a photo of every page in that report. Often we just get one page of the report, or we only get even half of a page, or people only take a picture of this main table that gives them their main variant. And DYRK1A researchers really want to have all of your variant information to truly start understanding DYRK1A syndrome. So please upload your reports to RareX. Sorry, this kind of ends with a call to arms, but there's over 800 families worldwide. So far, last I saw, there were about 60 families in RareX, and of those, we have about 12 genetic reports that have been uploaded to RareX. I know many of you have uploaded genetic reports to other platforms, such as the Matrix. I'm with Combined Brain, we use the Matrix, or have sent them directly to researchers. But if you could upload them to RareX, we're really trying to collate all of this data in one centralized base so that the DYRK1A International Alliance can work together with our collaborating researchers to really understand these genetic variants. Oh, and there's a link where you can sign up. It's pretty simple. All right, and then on to Anna. Anna is going to talk about actual research that we've been working, Combined Brain, we've been working together with the DYRK1A community, where these genetic variants actually really come into play along with the blood samples that were donated last year.
Okay. Kellen, did I kick you off? And now, okay. Can people see my screen? Okay. All right, cool. So I can't see you all, but okay. Good job, Kellen. That was great. Also, nice touch, Amy. I liked the recording of uploading the genetic report. Okay, so as Kellen said, I am going to spend the next probably 10 to 15 minutes giving a very general introduction to biomarkers. So primarily what they are, why do people keep talking to you about them, what do they do, some examples of biomarkers in the context of other neurological diseases. And then some preliminary biomarker data that came from DYRK1A patient samples themselves. Okay. Okay. All right. So in order to really talk about biomarkers and to understand the importance of biomarkers, I think it's really useful to show you their role and their place in the entire drug development pipeline for a rare genetic neurodevelopmental disorder such as DYRK1A syndrome. So, okay, I could see Kellen's pointer, so I'm assuming you can see my mouse over here. But if you look on the left-hand side of this blue line here, you can see that usually the first step in the drug development pipeline is you get the gene identified that is pathogenic, that's causing this sort of clinical phenotype or the symptoms observed in children or a certain population. And then usually one of the next things is that you need to develop either rodent or other animal models or cell models of the particular genetic variant or disorder so that you can start studying some of the molecular or phenotypic consequences of that genetic variant. So let's use DYRK1A pathogenic variants, for example. And I should say before I go on that the DYRK1A research community and foundation have a breadth of these resources already available. So there are patient-derived cell models as well as animal models, a variety of animal models actually that exist already for DYRK1A syndrome. Probably the next step in this pipeline is the importance of a natural history data collection program or a natural history study. And really, this is an observational study. So what this is just looking at the presence or prevalence of certain symptoms or features in DYRK1A syndrome and how they change over time. This is really important when you're thinking about designing clinical trial outcome measures because you want to pick outcome measures that are really relevant for the age at which you're designing your clinical trial for. And again, the DYRK1A community has, part of this, an aspect of this is filling out surveys, although they're not everybody's favorite thing to do, on various platforms and other clinical research opportunities. So people are really participating in this area as well. Another part of this is to identify the specific molecular mechanisms that are altered as a result of having mutations or variants in DYRK1A. And this is typically done by basic researchers, although to some extent can be done by clinicians as well. And as we keep going to the right along this timeline, you see disease concept, disease burden studies. And what this is really doing interviews with DYRK1A community members or families to, in an unbiased way, identify the symptoms that are most burdensome or impactful on families and their children. The reason for doing this is, again, to make sure that the outcome measures included in clinical trials are the ones that will make the biggest difference and impact on DYRK1A families. And then below, finally here, you see the identification of biomarkers. And we will talk a little bit more about what biomarkers are and why they're important here. But just briefly, these can be a variety of different patient-derived biosamples, and they're indicators of disease status. And I'll get more into that in a second. And then finally, on the right-hand side here, we have patient-centered outcome measures. So outcome measures, again, that matter are most impactful on patients and their families, sort of safety testing. And then all the way on the right-hand side, you have your first round of clinical trials and FDA approval of various drugs or personalized therapies being approved. Okay. So for the next handful of slides, though, I'm really going to focus on this part in the middle that is a critical part in the drug development pipeline, which is the identification of biomarkers. Okay, so I feel like a lot of people use this term and throw this term around. So I want to just sort of focus in and give some examples of what these most commonly are and why they're important and why the community is actually critical for identifying biomarkers for DYRK1A syndrome. So at its definition, a biomarker is an objective measure which can be used to either predict disease status, so whether someone is getting worse, someone is getting better, and or even sometimes it can be used to predict how an individual will respond to a particular treatment, some sort of experimental treatment, new medication, etc. Typically, when we think of biomarkers, we think of things that are biologic in nature, things that are coming from fluids, etc., or physiologic. Examples of things that are biologic biomarkers would be things like mRNAs or genes, epigenetic changes, so these would be small changes, structural changes above the level of DNA or mRNA. Proteins, which is an example of what we will show you in a little bit later, metabolites, and microbiota. These are all things that could potentially be biomarkers for DYRK1A syndrome. And where you can find these specific biologic biomarkers is you can find them in fluids and patient fluids, such as cerebrospinal fluid, which is very invasive to collect, but some of these considerably less invasive fluids and samples. So you can find biomarkers in blood, urine, saliva, as well as stool samples. And then the other probably most common biomarker, especially in the context of neurodevelopmental disorders that have epilepsy associated with them, are physiologic biomarkers such as electroencephalograms or EEGs, or something called an event-related potential, or ERPs. Now, as I'm sure many of you that are watching or listening are familiar with, EEGs are a measure of electrical activity in the brain. And here's a little cartoon of what this looks like. It's most commonly a cap that you put over the participant's head. It has a lot of electrical wires, and it allows researchers or clinicians to sort of monitor the electrical activity in the brain. Alterations in these electrical signals, which it's either due to a seizure, which you can detect very sensitively, or just other sort of novel, unique features of a neurological disorder. These can be used as a biomarker for example, DYRK1A syndrome. So here is an example of biomarkers in the context of another neurological disorder. And in this case, I used biomarkers for Alzheimer's disease, which I'm sure everybody is very familiar with. So biomarkers change. They are by nature biomarkers because they change with disease and they change with age. So probably two of the most well-known biomarkers in the context of Alzheimer's disease are things that you can measure in the cerebrospinal fluid as well as the blood or plasma of Alzheimer's disease patients. And these are neurofilament, short for NFL, or tau proteins. So these cluster of four figures right here, I won't focus on them a whole bunch, except for if you look at this figure on these bottom two figures right here, what you can see is that on the y-axis right here is neurofilament levels, and then on the x-axis here is age. And for this figure on the right-hand side on the bottom here, this is glial fibrillary acidic protein, or GFAP, which we'll talk about later. And then again, age on the x-axis here. And the only thing that I want to point out is that the levels of these two proteins increase consistently with age. And in this case, age is an indicator of disease progression. So as the disease gets worse or more severe, the levels of these two plasma proteins go up. So we can use this as an indicator of whether a treatment at some point might actually stop the increase in the level of these blood-based biomarkers. So this is in the context of Alzheimer's disease, but you can imagine that we could look at this in the context of an aging DYRK1A syndrome population. And this is an example of that. Okay, so to explain a little bit more about physiologic biomarkers, which is something that I think is going to be really important in the DYRK1A community. So as I said, electroencephalograms can be diagnostic. They, in a clinical setting, are often used to detect whether an individual is having seizures or other epileptic activity. But they can also be used to discover that there are unique EEG features that could be used as a biomarker for particular disorders. So here, there's a little cartoon or an example of EEG tracings. And you can see highlighted regions here that look unique to the rest of the tracings. And if you gather a bunch of these, you might start to notice that there are similar unique features in DYRK1A patients compared to unaffected patients. Theoretically, these features could be corrected by specific treatments or future treatment modalities in clinical trials. So this is something that is very correctable and could be sensitive to certain future experimental treatments. And these green figures here, these are examples of event-related potentials, which I mentioned on one of the first slides here. And the areas that are less green, whether they're in orange or blue here, they're a way of checking for differences in how the brain responds to certain stimuli. So an individual who's wearing the EEG cap still, they show a picture, they play a sound, and then they look and see what areas of the brain respond and how well do they respond. These can also be used for as examples of physiologic biomarkers that could be corrected with some sort of treatment. Okay, so the last handful of slides here, I think are probably for me the most exciting. And this is actually an example of biomarker data that we have generated from samples that we collected from the DYRK1A community. So these are blood samples that we have collected from DYRK1A patients as well as unaffected siblings to generate, I think, probably one of the first plasma proteomic data sets, so as a pilot biomarker project for DYRK1A. So what you're looking at right here is a figure of the sample breakdown for all of the disorders that we actually enrolled in this project. So our goal for this project was to get 20 plasma samples from a variety of neurodevelopmental disorders. And if you look at this figure, you can see the names of the disorders here across the x-axis, and then the number or the count of patients that had blood samples collected. And this blue arrow right here shows that we enrolled almost exactly 20 or collected blood samples from approximately 20 DYRK1A syndrome patients, and then about 10 unaffected sibling samples as well. Those are the red bar, and then the patient samples are the bluish purple bar here. So we had an excellent turnout from the DYRK1A community. And so what we have found, and this is a little confusing here, but the main takeaway message is that if you remember, we collected samples from over 30 neurodevelopmental disorders. One of those was DYRK1A syndrome. Now, what you're looking at right here, there are two clusters here. And I'm not going to spend a lot of time on the relevance of these two clusters, but what I want to tell you is that disorders are differentiated by color. So all of the different disorders are a different color dot here, and each dot is a participant. So if you look at DYRK1A right here, it's light blue. But the main takeaway message is that all of the disorders and all of the colors and all of the dots are pretty much interspersed, and there's no clear distinctive pattern. So there are no clear differences in plasma protein by these different neurodevelopmental disorders. And so because of that, what we wanted to do is we took all of the affected patients in all of these 30 neurodevelopmental disorders, and we said, okay, compared to unaffected samples, what proteins are changed? And when you look at that, so here is something that's called a volcano plot, which many of you might be familiar with, but the way that you read it is that individual dots here, individual red dots, if it's red, it indicates that that protein was significantly upregulated in, for example, DYRK1A patients. And there are 154 of those. So 154 upregulated proteins in DYRK1A syndrome. And then on the left-hand side of this volcano plot, you see the individual blue dots right here, which indicate that there are 57 significantly down-regulated proteins in DYRK1A patients. So there is a lot going on here, but one of the things that really jumped out, just based upon, for example, the biomarker work that's been done in other neurological disorders, is that you can see this protein, GFAP, or glial fibrillary acidic protein, is one of those proteins that is significantly upregulated in DYRK1A as well as other neurodevelopmental disorders. So what we wanted to look at was, okay, well, how much is this GFAP upregulated in DYRK1A? And the exciting thing about this is GFAP is, if you remember, that is a known biomarker for other neurological disorders. So it's already kind of exciting because of that. On top of that, it seems like, based on the literature, there could be some molecular connections between DYRK1A and this glial fibrillary acidic protein, which, by the way, is as a marker of astrocytes in the brain. Okay, so this is also kind of a heavy slide, but I think it's really exciting. So and I think it's my last one, so I'm gonna spend a couple minutes on this. So what we're looking at right now is actually just zooming in and only looking at protein levels for this glial fibrillary acidic protein. And the first line right here where my marker is hovering over right now, these are control samples. These are the sibling samples from our project right here. And the average for those control samples is sort of marked right here by this red line running vertically through our screen. And then below this, you can see the GFAP level for all of the neurodevelopmental disorders included in this project. And if you look about 9 or 10 lines down here, you can see these are DYRK1A syndrome samples, right? And you can see that the GFAP levels from the patients with DYRK1A syndrome is probably one of the most elevated compared to the other neurodevelopmental disorders included in this data set, which to me is a really good indication that this is something that we should probably pursue and dig into as a potential biomarker that is relatively unique to DYRK1A syndrome patients. Okay, so I lied, I had one more slide. So what does this mean? I think this means that there's reason to get really excited about some of the preliminary potential proteomic biomarkers that we found in this first pilot study. We certainly need to engage the community and collect not only samples from new patients, new participants from a broad age range. But it's also really important to get participants who have already donated samples, because we would be really interested in understanding how plasma protein levels change with age, right? Biomarkers change with age. So we at Combined Brain make it really easy, or we try to make it really easy to participate in biomarker projects like this, as well as a variety of other projects. We do collections. This is kind of a cartoon map of where we do collections that help generate the type of data we just talked about. You can speak with your clinician for helping collect samples, and then also we can help coordinate with the DYRK1A Foundation home visits as well for this type of stuff. Okay, I think that was the last slide. All right, I will stop sharing. Okay. Any questions? Questions in the chat?
Jane raised her hand. So Jane, if you want to come out of, Janet, if you want to come out of mute and ask your question.
Yeah, thanks. Sorry, I'm looking at one screen and I think the camera's on the other one, so apologies. So I'm in the UK. Are we able to help with this study? We're having a UK meetup. Is that an opportunity to get some of these samples?
Yeah, we've had some success. It's certainly a little bit more difficult than doing it at places in the US, but if we have enough lead time and enough interest, we can definitely coordinate international collections. It usually requires working with a researcher who already collects these types of samples, who can sort of help us while we're not over there. But it's definitely possible. It just requires a little bit more finagling.
Right.
So maybe the next
Maybe
step would
get in touch
be
direct.
just to better assess, how many folks would be interested in donating samples, and then also identifying a researcher. So maybe we can, start working on that.
Yeah. Yeah.
Brilliant. Yeah, thank you.
And their meetup is the last week of July. So it's gonna, since there's a date already, I think we have like a timeline that we can work with.
Oh, that's perfect. That's enough. That's plenty of time. That would be cool. Yeah, I'm happy to help.
Have Jennifer raise her hand. Jennifer, you can come out of mute.
Yeah. Hey, I had a question. So to get the biomarker research and the blood samples and all that to the correct person in the States, I live in Houston, Texas. Would I have to drive to one of those sites to get it collected? Or is there a way to coordinate it with a bunch of us that live in the Texas area? I think there's a handful of us.
So that's kind of a good question for you now. And we have our US conference happening in Texas in mid-July this year, right? And
I'm at another conference at the same time, unfortunately. So you can't go to that one.
So there's a couple ways to do it, right? You don't have to go to one of those sites that I sort of listed on that the United States cartoon. We've had a handful of groups do sort of like mini meetups where they have 5 or 6 families, and we sort of do it at someone's house, or we like get a conference room or something like that. The other thing, though, if it's like on a one-on-one basis, is we often, this is typically for really severe kids, but lots of groups use it, is we send nurses or phlebotomists to people's homes. And so that could be just on a per patient or per family level, or you can get 2 or 3 other families to come over and just do it at one individual's house.
Okay.
Those have been pretty successful. Yeah.
next question, we see Dhruv. I don't know if I pronounced your name correctly.
So that is my son's name actually. I'm from India, so my son is actually diagnosed with DYRK1, and the awareness is too less here. So if we upload the report in the RareX, so can we also get the guidance, like how is it separate from others, or is it similar, or is he having some other issues as well? Like when we are seeing other DYRK1 kids, we have a lots of similarity with Dhruv as well, and seizures also onset for him like last year. With the medication it has gone fine, so we see a lot of things which are very common, but we want to just see, like, okay, if this is the same which you have observed with the 20 other people.
Kellen, you want to respond to that?
Yeah, you could reach out to the DYRK1A Syndrome International Alliance, and we can, connect you. I mean, Oliver knows how to look at this as well, or I could look into it, or you could be one of us will assign ourselves to look in to your mutation and maybe give you some basic statistics on the frequency of your mutation, where the mutation lies in the protein, what kind of mutation it is, how common or uncommon it is. And research on that is kind of still ongoing. I can also point you to a bunch of research papers where people have looked into this. But yeah.
And also,
I just
if you
post here.
I was going to say, if you upload your genetic report to RareX, they will curate it for you. It takes a couple months, but if you upload it, you should, in theory, get a document that sort of explains a lot of the questions that you had. And they said it takes a couple months, but you will eventually get that.
That is great.
And
Yeah,
I put
I
the
would
organization. So in fact, like I put the organization email on the chat. So anyone that doesn't know how to get in contact with us is admin@DYRK1A.org. We'd be happy to kind of get the question where it needs to go.
Yeah, and I would say that that's sort of the importance of the RareX platform. So besides sort of uploading your genetic report and having that curated so that, we can sort of better understand the community, what mutations and differences that individuals may have, there's a series of surveys that are also part of RareX that we have that will, if you answer those, that'll help us better understand just what kind of symptoms different individuals might have with different mutations. And Anna did a great job of mentioning the importance of a natural history study, and so that's a lot of times where you can pull that data for something like RareX. So that serves almost as like a natural history study, because as you complete these surveys, that a lot of them are clinically validated, that when and if a treatment becomes available, you can leverage that data set in clinical trials, so that you don't necessarily have to run control groups where there's, usually in clinical trials, you have a control group that, maybe not receive the treatment. But in rare disease populations where there's not many individuals, it's not always feasible to run randomized control trials. So that's also the importance of, filling out some of the surveys to understand the natural history, aside from, the clinical diagnosis.
And one more thing which I heard here, like since the DYRK1 patients are very, very less, so the motivation for research is not that great in this particular field. Is that true? But when I saw in the internet, for example, artificial intelligence tools also, they said that DYRK1 is also responsible for many other diseases. So this is something where people are interested to have research on. So I'm really confused. Like for my kid, I wanted to know if there will be some research or some extensive work already going on, or is it like slowly going on because the number of people are too less. And my geneticist told like there might be several people who are having this disease, but they don't even know because genetic testing has recently come.
Yeah, those are all very accurate, I think, responses. I think, you know, in rare diseases, especially one like DYRK1A syndrome that does not affect as many individuals, although to your point, it's difficult to get a diagnosis because much of the time you need something like whole exome sequencing that not everybody has access to, you know, in order to get the diagnosis. But what we've seen in the past several years alone is that, you know, the research has significantly progressed and improved, and, you know, the knowledge that we have gained has been considerable. And that's why it's important to, you know, participate in some of these research studies, especially some of the biomarker studies, because as we generate patient samples, that can supplement what they're doing in the lab with, you know, animal models and cells and things like that. You can only go so far with those model systems. And so having, you know, patient data, survey data, including patient sample data is a very important aspect to that. And so that's our goal, you know, from the Medical Scientific Advisory Board and our community, as well as how can we, you know, leverage the community, generate, you know, good clinical data that other researchers and even pharmaceutical companies are going to be interested in collaborating with, because now we're sort of creating that database that they're going to be interested to study DYRK1A syndrome.
Thank you.
Q, we are on top of the hour. Can we get one last question? How is your time, Oliver, Anna?
Yeah, I'm good.
I'm good.
All right, the
Can stay
next
around.
one is Craig. You can get yourself out of mute and ask a question. Craig McIntosh.
Hey, Kellen and Anna, thanks for the talks. They were great. I just wanted to ask a question. I think you referenced the paper that I might have read about the haploinsufficient mice having a larger population of astrocytes in the developing brain and a higher, obviously, then GFAP. I think from that they found that the older developing mice had a larger difference. Did you guys sample enough patients to see if there was any stratification of age or sex based on GFP, or did you just not have enough samples? GFAP, sorry.
Yeah, so we didn't do a formal power calculation, but what I can tell you is that we had about 20 DYRK1A patients. And when you try to see if there's a relationship between GFAP and age, you don't see it in 20. You do see it when you start including other disorders. And so that to me thinks that we just had a sample size problem. No clear difference between males and females, but again, we didn't have a pretty even distribution. It was like 6 and 14 or something like that. So that also wasn't a very fair comparison. But at this point, no reason to think there's a sex effect.
All right, thank you.
So what was the total, when you included like all the other disorders, what is the total sample size for that you were able to detect an effect on age?
Like 80. But
Yeah.
I haven't gone, I mean, I did it once with that number, just age, sex matched, other neurodevelopmental disorders. I didn't pick by, you know, ones that were other kinases or something like that. So I also didn't go back and see how few would I need to do that. So don't think you need 80 for this. Yeah. But I do think from like the level of should someone dig into that and look at it using some of the DYRK1A patient-derived models we have, I think that would be really cool.
So thank you very much, all that participate. I know we didn't get all the questions. Please forward your questions to admin@DYRK1A.org and we can appropriate answer and get that back to you. So the ones that have questions in the chat, I will collect those questions and we can get back in reason of time. So I really appreciate and can't wait for you to join us for our next research roundtable in a few months. And yes, the record will be available in our YouTube channel. give me like a day and I will post it there. Also, everybody's going to receive a survey. If you please answer the survey, that was going to guide us for the next events.
Cool.
Thank
Thank you, everyone.
Thank you, everyone.
Thanks, everyone.